Animal cells. There are 5 parts to know in an animal cell. We call these organelles. The nucleus controls the cell and contains your DNA, genes, and chromosomes. The cytoplasm is the jelly-like substance where chemical reactions happen. The cell membrane controls what goes in and out of the cell, a bit like a bouncer on a door. Mitochondria release energy through aerobic respiration. So the more active a cell is, the more mitochondria it has. So think of something like a sperm cell that has to swim about. It needs lots of mitochondria to provide energy. And finally, ribosomes. Ribosomes make proteins. So think about the keratin in your nails and also your hair. For the exam, you need to be able to label all 5 and say what each one does. A plant cell has everything that an animal cell has, plus 3 extras. Firstly, a cellulose cell wall for support. Think about something like celery that's really quite tough. Well, that's largely due to the cellulose cell wall. Chloroplasts, where photosynthesis happens, they're full of a pigment called chlorophyll, which is why they're green. Don't say chloroplasts are in every plant cell. For example, root hair cells don't have any chloroplasts because they're underground where it's dark and they don't photosynthesize. There's no light there. Examiners love asking that question. And finally, a permanent vacuole filled with cell sap that keeps the cell turgid. When we think of turgid, we're thinking of something tough and a little bit swollen. Now we move on to the light microscope. A light microscope shines light up through a thin specimen. So our specimen might be something like a skin cell, or a cheek cell, or a plant leaf cell. You'll remember having seen a demonstration or done an investigation where you've taken a really thin slice of onion and put it under the microscope. Well, that specimen, that onion, was really thin so that light could get through it. In a microscope, you have 2 lenses that do the work. The eyepiece lens at the top, which is where you put your eye and look down, and the objective lens, and that's the one that you can swing carefully into place over the specimen. Because there are 2 lenses, we need to think about the total magnification using the eyepiece times the objective lens. A 10 times eyepiece with a 40 times objective lens gives you 400 times magnification. So just remember that when you are doing the calculation for magnification using a microscope, there are 2 lenses, and therefore you have to think about the eyepiece lens and you multiply it by the objective lens to give you the total magnification. Light microscopes go up to about 2,000 times magnification. That's quite big. However, the magnification is not powerful enough to see really tiny objects, and that's where we use electron microscopes. They go up to 2 million times magnification and with a much higher resolution, and that resolution allows you to see an object more clearly. But the problem with electron microscopes, apart from being so expensive and generally very big, is that they can't show living cells. The cell has to be dead first. That's a quick comparison that the examiner often asks for. Enzymes. Enzymes are biological catalysts. That just means they speed reactions up without being used up themselves. Your body uses them to build things like proteins and to break things down like the food that you eat. Each enzyme has a special section called the active site. The molecule that the enzyme works on is called the substrate, and it slots into the active site like a key into a lock. And that's why we call this the lock and key model. 3 things affect how well an enzyme works: temperature, pH, and substrate concentration. If the temperature is too high or the pH is too far from the optimum, the active site changes shape permanently, and that means the substrate can no longer fit. When that happens, we say that the enzyme has denatured. An examiner tip: never say a denatured enzyme has died. Enzymes were never alive. You say the active site has changed shape and the substrate no longer fits. Diffusion. Diffusion is the net movement of particles from a high concentration to a low concentration down a concentration gradient. Because it's down a concentration gradient, think of it a bit like a ball rolling down a hill, it's passive and you don't need any energy to do it. Some examples are oxygen diffusing into your cells and carbon dioxide diffusing out of your cells. The bigger the gradient, the faster the diffusion. Higher temperatures also speed up diffusion because the particles are moving about much faster and are more likely to bang into each other. That's a nice link to kinetic energy in physics. We can't talk about diffusion without referring to the special type of diffusion in liquids, osmosis. Osmosis is the movement of water across a partially permeable membrane. It allows some things through and it doesn't allow other things through. Think about the organelles that we just identified. This means that particles will move from a dilute solution to a more concentrated one. And this often trips up students. Dilute solution, does that have a lot of water or a little bit of water? If you think about a glass of Ribena, if it's dilute, you add lots of water to it. If it's concentrated, it doesn't have much water in it. So osmosis is where we move from a dilute solution with lots of water to a concentrated solution which doesn't have lots of water. Again, it's down a concentration gradient and it does not require energy. When there is a higher concentration of water outside the cell than inside the cell, water moves into the cell by osmosis down a concentration gradient. In plant cells, the vacuole swells and pushes against the cell wall, and this makes the cell turgid. This is not a bad thing, it helps support the plant and keeps it upright. In animal cells, water also moves into the cell, but because animal cells don't have a cell wall, they can swell and burst. When there is a lower concentration of water outside the cell than inside the cell, water moves out of the cell by osmosis. In plant cells, the membrane pulls away from the cell wall and the cell becomes plasmalized. This can cause plants to wilt. In animal cells, the cell shrinks because water leaves the cell. The way to remember this is if you're sitting in a bath which has lots of water, you end up getting wrinkly fingers. Active transport moves particles against the concentration gradient, this time from a low to a high concentration. This takes energy. The clue is in the name, active. So it requires ATP from respiration. And remember, respiration happens in the mitochondria. The classic example is root hair cells absorbing minerals from the soil. Plants need mineral ions such as nitrates and magnesium for healthy growth. For example, magnesium is needed for chlorophyll. Without it, the leaves of a plant look a bit yellow. At the root hair cells, there is a lower concentration of these mineral ions in the soil than inside the root hair cell already. The plant needs to move mineral ions from a lower concentration in the soil to a higher concentration inside the cell, and this is against the concentration gradient. The plant uses active transport, and this requires energy from respiration to move the mineral ions into the root hair cell. Cells in the small intestine absorb glucose from digested food into the bloodstream. There is a lower concentration of glucose in the small intestine than in the blood already. This means glucose needs to move from a lower concentration to a higher concentration. The cells therefore use active transport, which requires energy from respiration to move glucose into the blood. When we look at transport surfaces, we need to refer to surface area to volume. Smaller objects have a larger surface area to volume ratio, and that means substances can diffuse in and out fast enough. So if you have a very small object and it's respiring, it can easily get oxygen into it. The oxygen does not have far to travel. However, as organisms get bigger, that ratio drops. So they need specialized exchange surfaces. And that might be lungs for gas exchange, gills in fish, or the small intestine for digestion. All of them have huge surface areas. They also have thin walls and a good blood supply to carry gases. A quick exam tip: if you're asked to calculate, work out the total surface area, then the volume, and then divide. Cell division, and we look at the first, which is mitosis. Mitosis produces two genetically identical daughter cells. Before it starts, in interphase, the cell copies all of its DNA, and it starts with copying because it's going to end up with two copies of the original cell. Each of those cells needs to have one of its own copy. So you double the amount so that when it splits into those two daughter cells, each daughter cell has its own full copy of chromosomes. Then there are 4 stages: prophase, metaphase, anaphase, and telophase. And the mnemonic for that is IPMAT, IPMAT. After IPMAT, you have cytokinesis, where you actually have the splitting of the cytoplasm. Stem cells and growth. Stem cells are unspecialized cells, and there are 2 types. Embryonic stem cells, the ones found in an early embryo, can become anything. They can become a liver cell, a nerve cell, a muscle cell. This is how a single fertilized egg manages to build an entire human being. Adult stem cells are more limited. The ones in your bone marrow can make different types of blood cell, but they can't make nerve cells and they can't make muscle cells. Plants have stem cells too. Plants keep growing for their whole lives. Their growth happens at specific points along every root and shoot. Cells divide in a region called the meristem, and that is at the tip of a root or the tip of a shoot. Just behind that, the cells elongate, so they get longer. And further back still, the cells differentiate into specialized types such as xylem, phloem, or root hair cells. There are 3 uses for human stem cells that you need to know about: treating leukemia with bone marrow transplants, repairing damaged nerves, and growing new tissues to replace damaged ones. There are also 3 risks or downsides to weigh against those benefits. Firstly, some people object to using embryos on ethical grounds, so they don't think it's right. Secondly, the patient's immune system might reject the new cells. And thirdly, there's a small risk of passing on infection. The nervous system. Your central nervous system, or CNS, is made up of your brain and your spinal cord. Everything else, all the nerves going to and from it, makes up the peripheral nervous system. Periphery means around the outside. Nerve cells, which we also call neurons, come in three types. Sensory neurons carry electrical signals into the CNS from receptors. Receptors are cells that detect things, and they're found in your sense organs. So light receptors in your eye and sound receptors in your ear. Motor neurons carry the signal back out of the CNS to an effector. An effector is whatever actually does something in response. And there are two types. You can either have an effector which is a muscle, and that's going to contract to move, like an arm muscle that yanks your hand away from a hot pan. Or an effector could be a gland, which releases a chemical, like your adrenal glands releasing adrenaline when you're scared, or your salivary glands releasing saliva when you smell food. One important detail, neurons don't actually touch each other. They meet at a tiny gap called a synapse. The signal can't jump the gap as electricity, so the neuron releases a chemical called a neurotransmitter that crosses to the next neuron and gets the signal going again. A reflex is a very fast response that bypasses the brain completely. It uses a third type of neuron called a relay neuron, which sits in the spinal cord. That's why reflexes are rapid, automatic, and involuntary. Those are three words that examiners love to see. The classic reflex example is stepping on a sharp pin. Your foot jerks back before the pain even registers. But there are more. Blinking when something flies at your face, your pupil shrinking in bright light, coughing when something goes down the wrong way, and also the gag reflex. All of them are rapid responses that protect you before your brain catches up. The order of a reflex arc is what gets you the marks. Stimulus, receptor, sensory neuron, relay neuron, and motor neuron, effector, response. Your sensory neuron comes first before your motor neuron. Something else to be careful of, do not say the brain tells the muscle to move for a withdrawal reflex. The spinal cord acts first and the brain catches up later. Remember, sensory goes in, motor goes out. Growth and differentiation. Growth works differently in animals and plants. In animals, cells divide by mitosis and then differentiate, meaning they take on a specific job, such as a skin cell, muscle cell, or blood cell. Doctors track how children grow using percentile charts. If you're on the 50th centile for height, that means half the children your age are taller and half are shorter. In plants, growth happens at specific points along every root and shoot. Cells divide in a region called the meristem. Remember, plants divide, elongate, and differentiate. They have three zones working back from the tip. Make sure you know the difference between differentiation and cell division. Differentiation means becoming specialized. Cell division means making more cells. They're different things and they happen in order. Divide first, then differentiate. DNA structure. DNA is the chemical that carries your genetic information. It's a twisted ladder shape called a double helix. The two sides of the ladder are made of sugars and phosphates. The rungs across the middle are made of pairs of bases. There are only 4 bases, and they always pair up in the same way. A pairs with T and C pairs with G. The order of the bases along the ladder is what determines the protein that is made. Different orders produce different proteins, and different proteins produce different characteristics. A gene is simply a section of DNA that carries the instructions for making one particular protein. Inheritance. You inherit genes from your parents, half from your mum and half from your dad. For each gene, you have 2 copies, called alleles, and you have 1 from each parent. So alleles are a pair of genes. Alleles come in 2 types. You have dominant alleles, which are written with a capital letter, such as big B, and they always show their effect. You only need 1 copy and they will show up. Recessive alleles, on the other hand, are written in lowercase, such as small b. They only show their effect if you have 2 copies, and that's 1 from each parent. The characteristic you can actually see, like brown eyes or blue eyes, is called your phenotype. So what you look like. The combination of alleles such as big B, or small b, so the letters, is called your genotype. A Punnett square is a simple grid used to predict which alleles offspring could inherit from their parents. If both parents carry the genotype big B, small b, there is a 3 in 4 chance of a child showing the dominant characteristic, and a 1 in 4 chance, or 25%, of showing the recessive characteristic. Family pedigree diagrams also appear in exams. These are family trees used to track how a genetic condition passes through generations. Someone who carries a recessive allele without showing the characteristic themselves is called a carrier. Variation is simply the differences between individuals of the same species, and it comes from 3 sources. Genetic variation is the stuff that you're born with, like your eye colour or your blood group. Environmental variation comes from things that happen during your life, like scars or the language and the accent that you develop. And many characteristics, like height and weight, come from a mix of both. They depend on the genes you inherit and what you eat and how active you are, for example. Variation also appears in 2 shapes when plotted on a graph. Continuous variation, where any value is possible, like height, gives you a smooth bell curve. Discrete variation, where individuals fall into distinct categories, like blood group, are you A, B, AB, or O, this gives you a bar chart. So remember, a bell curve is continuous and a bar chart is discrete. Sex determination. Your biological sex is determined by 2 of your chromosomes, called the sex chromosomes. Females have 2 X chromosomes, written XX. Males have 1 X and 1 Y, written XY. Your mum always passes on an X, because that's all she has. Your dad can pass on either an X or a Y. So it's actually your dad who determines the biological sex of the baby. A 50-50 chance either way. Meiosis is the type of cell division that we use to produce gametes, sperm cells in males and egg cells in females. Unlike mitosis, the cell divides twice, producing 4 daughter cells instead of 2. Each new cell contains half the normal number of chromosomes. With this many changes, of course, the daughter cells are not going to be identical to the parent cell. These cells are described as haploid because they contain a single set of chromosomes. Think haploid, half. Body cells are diploid because they contain two sets of chromosomes, one set from your mum and one set from your dad. This reduction is important because during fertilization, the sperm nucleus and the egg nucleus join together, restoring the full number of chromosomes. The fertilized egg is called a zygote, which then divides by mitosis as it develops into an embryo. So to be clear, mitosis produces two identical body cells. Meiosis produces 4 different gametes. Mitosis is for growth and repair, and meiosis is for making gametes, so egg and sperm cells. Mitosis has 1 division and meiosis has 2 divisions. Meiosis creates genetic variation because every sperm cell and every egg cell contains a different combination of alleles. That's why brothers and sisters often look similar but are not genetically identical. Identical twins are the exception. They develop from a single fertilized egg that splits into 2 embryos. Natural selection. Natural selection is the process that Charles Darwin used to explain how species change over time. Individuals within a species are not all identical. There's variation, and some individuals are naturally better adapted to their environment than others. Environmental pressures such as predators, disease, competition for food, or changes in climate affect which individuals are more likely to survive. Those with advantageous characteristics are more likely to reproduce and to pass their alleles on to the next generation. Over many generations, these useful alleles become more common in the population, and the species gradually evolves. A modern example is antibiotic resistance in bacteria such as MRSA. Antibiotics kill most bacteria, but some may already carry a mutation that makes them resistant. Those bacteria survive, reproduce rapidly, and pass the resistance allele on, creating strains that are much harder to treat. In biology, the phrase survival of the fittest means best adapted to the environment, not necessarily the strongest. To recap, there are 4 steps in natural selection: variation, selection pressure, survival, reproduction. There are 2 common misunderstandings about evolution that often show up in the exam. The first is that individual organisms evolve during their own lifetime. This is not the case. Evolution happens across populations over many generations. So a giraffe does not stretch its neck and pass that change on to its offspring. Instead, some giraffes are naturally born with slightly longer necks. If that characteristic helps them survive and reproduce, the alleles for longer necks become more common over time. The second misunderstanding is that evolution has a final goal or direction. Species are not trying to become perfect or better. Adaptations simply increase the chances of survival in a particular environment. If the environment changes, different adaptations may become advantageous instead. Evidence for evolution. There are several lines of evidence for evolution. Fossils. They show how organisms have changed over millions of years. For Edexcel specifically, you need to know about 3 key human fossils. RD, around 4.4 million years old. Lucy, around 3.2 million years old. And the much later fossils discovered by the Leakey family. The development of stone tools over time and the way archaeologists date them from the rock layers they're found in also counts as evidence of human evolution. Antibiotic-resistant bacteria show evolution happening in real time within decades. And this is because they divide and grow so fast. And modern DNA studies reveal close genetic relationships between species. If you're asked for evidence, give at least 2 different types. Naming Ardy or Lucy is a strong Edexcel specific answer. You can think of fossils for the deep past, stone tools for human evolution, antibiotic resistance or DNA for right now. Classification. Classification is simply a way of sorting living things into groups so that we can study them. The old system was invented by a scientist called Carl Linnaeus, so we call it the Linnaean classification system. It has 7 ranks, from the biggest group to the smallest. Kingdom, phylum, class, order, family, genus, species. Every living thing fits somewhere in that hierarchy. The newest system uses just 3 big groups called domains. And we have bacteria, archaea, and eukarya. Eukarya is the domain that contains all animals, plants, fungi, and us. Scientists switched to this system because DNA evidence showed the old 5-kingdom system was not quite accurate. Every species also gets its own 2-word scientific name. Take our own Homo sapiens. The first word is the genus. The second is the species. Everyone in every country uses the same 2-name system, so there's no confusion across languages. For Edexcel, know that the change from 5 kingdoms to 3 domains happened because of DNA evidence. And learn the 7 Linnaean ranks in order. King Philip came over for good soup. Selective breeding and genetic engineering. There are 2 ways humans change the characteristics of plants and animals. Selective breeding is the traditional method. You choose the parents that show the characteristic that you want and you breed them together. Then you select the best of their offspring and you breed those. Over many generations, the desired characteristic becomes stronger and more consistent. That's how we got every dog breed from wolves and how we developed cows that produce far more milk than their wild ancestors. Genetic engineering. This is also called genetic modification, and it's the modern approach. Scientists take a useful gene from one organism and insert it directly into another. The most famous example is taking the human gene for insulin and putting it into bacteria, and then having those bacteria produce human insulin for people with type 1 diabetes. Selective breeding works through normal reproduction over many generations. Genetic engineering happens in a laboratory and is much faster, so make sure you use the right name for each. Pathogens. A pathogen is a microorganism that causes a disease that you can catch from other people or other organisms. Diseases that spread from person to person are called communicable. Not every microorganism is a pathogen. Many bacteria are harmless and some are genuinely useful, like the ones living in your gut. There are 4 types of pathogen: bacteria, viruses, fungi, and protists. For Edexcel, you need to know 7 specific diseases, and for each one, learn the pathogen type, how it spreads, and 1 main symptom. Cholera is caused by bacteria and spreads through drinking water contaminated with feces or poo. It causes severe diarrhea. Tuberculosis, or TB, is also bacterial and spreads through the air when an infected person coughs. It damages the lungs, and in serious cases, sufferers can cough up blood. Cholera ash dieback is caused by a fungus that infects ash trees. It spreads on the wind between trees, causing the leaves to fall and the branches to die back. Malaria is caused by a protist and spreads through mosquito bites. It causes fever and damages red blood cells. HIV is a virus spread through bodily fluids and unprotected sex. It attacks the immune system and, if untreated, can develop into AIDS. Stomach ulcers are caused by a bacterium called Helicobacter pylori, or H. pylori for short, and this is spread through contaminated food and water and causes stomach pain and internal bleeding. Ebola is a virus spread through bodily fluids, and that causes severe bleeding and fever. Two diseases to discuss are also sexually transmitted infections, HIV and chlamydia. Chlamydia is a bacterial sexually transmitted infection. Both spread less when people use condoms. Just an examiner tip, mosquitoes are not the pathogen for malaria. They are what's called a vector. A vector is the thing that carries the pathogen. The actual pathogen is the protist. So imagine a mosquito and inside it is a small pathogen called a protist, and that's what causes the infection or the disease. Body defenses. Before your immune system has to get involved, your body has a range of non-specific defenses. Non-specific simply means they work against any pathogen and not just one particular type. Your skin is a physical barrier and most pathogens can't get through it. In your airways, mucus traps pathogens and tiny hairs called cilia waft the trapped material back up your throat so that you can swallow it. The hairs in your nose filter the air that you breathe in. Your stomach acid is strong enough to kill most pathogens you swallow with your food. Your tears contain an enzyme called lysozyme that breaks bacteria apart. And when you cut yourself, tiny cell fragments called platelets cause the blood to clot, forming a scab that seals the wound. So non-specific means it works against any pathogen, not just one type. And those are your first line of defense before the white blood cells get involved. The immune response. If a pathogen gets past the non-specific defenses, your white blood cells get to work. There are two main types and they do different jobs. Phagocytes engulf pathogens, basically eating them whole. Think of Pac-Man. And that process is called phagocytosis. Lymphocytes do something cleverer. Every pathogen has unique markers on its surface called antigens. A lymphocyte recognizes a specific antigen and starts producing antibodies. These are proteins that are exactly the right shape to bind to that antigen. Once bound, the antibodies mark the pathogen so phagocytes can come and destroy it. After the infection is gone, some lymphocytes stay behind as memory cells. If you ever encounter the same pathogen again, those memory cells recognize it, and the response this time is very fast, meaning that you don't get very ill. You might not even get ill at all. Vaccinations work by using this same system. A vaccine contains a dead or weakened version of a pathogen. Your immune system responds to it without you actually getting sick. It produces antibodies and leaves memory cells behind. So, if you meet the real pathogen later, you're already prepared. This is called the secondary immune response. If enough people in the population are vaccinated, The pathogen can't spread easily, so even unvaccinated people are protected, and that is called herd immunity. A precision point, vaccines do not usually stop pathogens entering your body. What they do is get your immune system ready to fight back so fast that the pathogen cannot take hold. Antibodies do not kill pathogens directly. They bind to antigens and mark them. The phagocytes do the destroying. And just to differentiate, antigens are on the pathogen, antibodies are from you. Medicines and antibiotic resistance. Making a new medicine is a long process with 4 main stages. The first stage is discovery. Scientists find a chemical that might work. Second, development. They adjust and improve it. Third, preclinical testing. They try it out on cells in the lab and then on animals. Fourth, clinical trials. They give it to actual humans. Clinical trials happen in 3 phases. Phase 1 uses a small group of healthy volunteers just to check that the drug is safe. Phases 2 and 3 use larger groups of patients who actually have the condition to check the drug works and to establish the right dose. Antibiotics are a special type of drug. They kill bacteria. They do not work on viruses because viruses hide inside your own cells where the antibiotic cannot reach. So no antibiotics will not help when you have a cold or flu. Antibiotic resistance is a serious and growing problem. Some bacteria evolve to survive antibiotics, and those bacteria can then spread. And that's why doctors only prescribe antibiotics when genuinely necessary, and why you must always finish the full course, even when you start feeling better. Safe and effective are not the same thing. Preclinical testing and phase 1 check that the drug is safe. Phases 2 and 3 check that it actually works. That's a very important distinction to make in your exam. Non-communicable diseases. Non-communicable diseases are conditions that you cannot catch from someone else. They build up over time and are usually caused by several factors working together, not just one. The World Health Organization says that health is more than just not being ill, it's a state of complete physical, mental, and social well-being, which means mental health counts too. The main lifestyle factors that raise your risk of non-communicable diseases include a high body mass index, or BMI, which is a measure of weight relative to height and is linked to obesity. Drinking too much, so alcohol, smoking, and also a poor diet. Each of these raises your chance of developing cardiovascular disease, type 2 diabetes, lung cancer, or liver disease. These conditions are also connected to each other. Obesity raises the risk of type 2 diabetes, and type 2 diabetes in turn raises the risk of cardiovascular disease. For cardiovascular disease specifically, that's problems with the heart and circulation, there are 3 types of treatment that you need to know about, from least to most invasive. First of all, lifestyle changes. More exercise, a healthier diet, and quitting smoking. Next is long-term medication, such as statins to lower cholesterol. And finally, surgery, such as fitting a stent to widen a blocked artery or performing a heart bypass. Examiner tip: if a question asks you to evaluate treatments for cardiovascular disease, cover all three: lifestyle, medication, and surgery, and weigh them against each other in terms of cost, success rate, side effects, and how invasive they are. So that's Edexcel Biology Paper 1 covered. I hope that it's helped you prepare for your exam, and good luck.